welcome to the breakfast at UMHealth. So my name is Fauzi. So I'll be your moderator for today. So today we have a very good lecturers and very promising lecturers. So we're happy to have Dr. Syed Mahmood with us here today. So he will present his talk on the progress of commercial drug delivery technologies. So before that, allow me to introduce Dr. Syed Mahmood. Dr. Syed Mahmood has completed his bachelor in pharmacy in 2011 and completed his master in pharmaceutics in 2013. He also holds a postgraduate diploma in pharmaceutical drug regulatory affairs. In 2014, he started his PhD and completed in 2017. His PhD research was on drug delivery and technologies. After his PhD, he worked at University of Malaysia, Pahang as a senior lecturer teaching in the pharmaceutical engineering department. His specialization is in the field of the pharmaceutical technology and engineering area, which include drug delivery, focusing on the transdermal delivery using microneedle and nanotechnology. He also interested in the in vivo animal model studies, analytical chemistry, including the LCMS and HPLC, wound healing studies, hydrogel and nanofibers, and system validation and qualification. Currently, he is working at University of Malaya in the Faculty of Pharmacy, attached to the Pharmaceutical Technology Department. So, without further ado, I would like to welcome Dr. Syed Mahmood to give his talks. So, for everyone who wants to have a questions, and then you can send your question to the chat box. Thank you very much. So, welcome, Dr. Syed Mahmood.
Thank you, Dr. Fauzi, for the introduction, and good morning, everyone. All are joining, and it is a pleasure to speak here on behalf of Breakfast for others. Okay, so I will, without delaying, I will share my screen first, Fauzi, and please confirm whether that the screen is visible or not. Yeah. Minimizing of the target accumulation and facilitating the patient compliance. Yeah. So as a therapeutic modalities, the expand beyond the small molecules to include nucleic acids, peptides, proteins, and antibodies. Drug delivery technologies were adapted to address the challenges that emerged. Okay, so I think we all are aware about all these terminologies, that is small molecules and nucleic acids and protein, peptide, and antibodies, that every day, or you can see that the medicinal products from these things are coming up and we are using them in our daily life. Okay, like for recent example is that people have used the
sorry, Dr. Syed.
Yeah.
Is it possible that you can make it your step to the slideshow, please?
So we all are aware about all these things, that what are small molecules, that the drug we are taking every day, this comes under the category of small molecules like paracetamol and other medications like antihypertensive medications and other kind of medications like anti-allergic and so on. And then we are also bringing in the protein peptides and all these things like insulins and so on. And then we came up with all this nucleic acid and all these things like this for our vaccine delivery and so on. So I mean, I would like to say in this thing that every day we are using all these type of molecules in our life, and we are very well aware about all these updates and about all these new drug delivery technologies that being addressed or that being introduced in our life these days. Okay, so the delivery strategies have greatly helped converting promising therapeutics into successful therapies. So the content will be the classes of therapeutics and delivery challenges with the examples. So I will be trying to cover up one by one all the therapeutic classes that we have, and then what are the challenges associated with them, and a perfect example, like what is the good example right now. And then I will try to identify the three drug delivery paradigms, that what are the three drug delivery things, or the associated problems, or what we can do with the drug delivery modification so that to achieve more suitable drug deliveries and so on. So it started how it started. So you can see that I will just give you a basic about the drug delivery or the generation of drug deliveries, that it started with the generation 1, that is everyone is aware what is generation 1, that is conventional capsules, tablets, emulsions, and all this thing. I think 90% of the medication that we are using comes under generation one, and I think uh, tablet, capsule, and all the liquid dosage forms, solid dosage forms are still categorized under this. Then we have generation two, that is a little bit modification and a little bit modified actions were there, like entry-coated, repeated, or prolonged release, and so on. Example of prolonged release or a sustained release is that we have a medication that comes for the diabetic control, okay? The people who are suffering from high blood sugar. So in that case, we have now medication that is only given one time, okay? So, and it can be used for a whole day, okay? So I think if here we have medical or a pharmacy student, so they can recognize this concept that we are going into this modification action systems, and this comes under generation two. Then we have generation three, that is the controlled delivery system, that is osmotically swelling, diffusion, and control delivery system. This kind of delivery system act according to your body type. Okay, so like suppose someone want to release the drug at the small intestine or at the lower part of the stomach, escaping from the pH and all this thing, So this comes under third generation, and then we have fourth generation of medication or the delivery system, that is targeted delivery system, that is the targeted, modulated, self-regulated delivery systems. In this one also you have nanotechnology aspects, that is transdermal delivery system, targeted delivery system, that is magnetite nanoparticles for targeting the cancer tumors and all this thing. Then we have fifth generation delivery system that is also flourishing, and but it is, it's still at the, you can say, it's still not yet practiced like generation three or generation two or like this, but still they are being highly, or they are being researched very actively. And you can see majority of the research in a very in universities is being targeted towards this thing only, that the gene therapy, long-term delivery systems, nanorobots, biomedical engineering, and so on. So the generation five is being studied very aggressively, and majority of the research right now is being done on generation five only. Okay, in terms of pharmaceutical technology, pharmaceutics, and also like molecular biology and all this thing. Okay, so these are some of the things that today I will be covering up. Um, that is the small molecules, protein peptides, and so on. Fauzi, how can I remove this one? This,
So remove what? Sorry, what is that?
yeah, this one, I have coming up on the, this one. I don't know how to remove this one.
Because so far from here, I can see your slides only. So
Ah, okay. I can. Okay, yeah, sorry. So in the slides, I will try to cover up all these small molecules and what are the challenges associated with them. That is small molecules, like controlling pKs, improving solubility, improving permeability, target development, reducing off-target toxicity. Same goes with the protein peptides, antibodies, nucleic acid, and live cell therapy. So these are the challenges that are associated with them. Then we will try to cover up the three paradigm of the drug delivery or the drug delivery system. That is the drug modification, environmental modification, and the drug delivery system. So in drug delivery system, you can see that there are so many examples of drug delivery. That is the starts with all this kind of delivery system, injections, wound healing, and so on. Okay. So the new generation of therapeutics include protein and peptides, monoclonal antibodies, nucleic acid, and live cell therapies have provided new therapeutic functions. The new functions brought about additional challenges, notably in solubility, instability, particularly for the protein peptides, and intracellular delivery requirements, especially for nucleic acid viability and expansion, like for live cells. So the drug delivery strategies have evolved to address the following challenges. So you can see that whatever research is being done in pharmaceutical technology field or even related to the pharmacy, the challenges and the concept stays the same. Like we are targeting and we are evaluating the activity of a drug. We are trying to make it more potent. We are trying to reduce increase the solubility. We are trying to challenge the pharmacokinetic and pharmacodynamic and all these things. So these are the things. So then only we can get a proper efficacy of the drug, so that while minimizing the side effects and while getting the higher therapeutic effect. So this is one of the major concept of pharmaceutical research, or you can say the pharmaceutical how we are modifying our drug delivery systems. Yeah. So in pharmacotherapeutics also we have that is high dose, adverse effects, and low patient compliance. Then let's start with the small molecules. I'll not cover up everything. I'll just try to be brief in this one so that we can still cover up with the time. That the small molecule drugs are were the primary class of therapeutics. Like everybody is aware about all this primary class of therapeutics, like what we are taking every day, like paracetamol, butanol, propanol, and all these things. These are the primary class, or you can say the small molecules. Okay, small molecules are like less than 900 Daltons. So the delivery of the small molecule is largely dictated by their physicochemical properties, which heavily influence the bioavailability of the drug. Delivery effort was focused on improving the solubility of the drugs, controlling their release, broadening their activities, and adjusting their pharmacokinetic. So the major problem comes with them is that for rapid diffusion and access of systemic vasculature, the small molecule must be freely soluble in biological fluid. Hence, this limit or hampers the active therapeutic utility of poorly soluble molecules. So of course, in small molecules, we have four classes of drugs, that is BCS classes, that is class 1, 2, 3, and 4. So these four classes are divided according to their solubility and according to their permeability, right? So according to this one, there are many challenges in delivering the small molecules. Some molecules cannot be delivered orally. Some can only be delivered through systemic circulation because of the high rate of degradation, high rate of effect of the pH, and so on. So, for example, so about 90% of the preclinical drug candidates are low solubility compounds. So this remains a challenge strategies to overcome the low bioavailability. So in this one, for example, the clinical success, such as the intervention of ciprofloxacin, which formulate with lactic acid to improve its solubility via pH modulation. So here the things is that any physician which are writing a small molecules to any patient, so also they want to see, or also they want to go up with the combination of medication. So these are also something in which primarily to introduce a combinational drug therapy, we need to see that if there is any potential toxicity or not, and all the toxicity level studies and so on, that starts with the preclinical and then finally going for the clinical one. Okay. Uh, okay, so what are the challenges associated with the small molecules? If you will see in the table down, that is the for controlling PK parameters, in particular half-life, biodistribution, exposure, and maximum concentration. So this is like PK parameter is very important for the drug, a particular drug. Then only it can be related whether it is successful or it is not successful. Okay. So all the scientists, those who are working right now in pharmaceutical technology field, they are preparing a specific or a controlled formulation just to overcome all these PK parameters. These PK parameters are half-life, biodistribution, drug blood concentration, and so on. Okay. So the example is osmotically controlled release oral delivery system of methyl phenidate hydrochloride to overcome the tolerance of the drug by controlling its PKs. Okay, and what are the PKs here we have? These are the PKs, that's half-life and Cmax, Tmax, and so on. Okay, and then we have another example, that is the improving the solubility of the small molecule, that is ritonavir, a protease inhibitor of HIV treatment, that is thiazole modified for improved metabolic stability and aqueous solubility. So what is protease inhibitor? That if somebody is already having a sign of HIV, or it can be a potent HIV case, so we are taking this protease inhibitor just to delay the HIV infection that it becomes to AIDS. Okay, so this is the class of this drug, that it is inhibit the HIV virus to spread more. Okay. So these are some of the examples, of course. Then we have peptides. That is, peptides are the chain of 2 to 50 amino acids, and the protein, that is chain of 50 and more amino acids, have evolved with the human body to have excellent selectivity for a specific protein targets. Their large size and diverse tertiary structure increase the points of contacts with specific protein pockets, granting the peptides and protein higher potency and reduce toxicity than many small molecules. So of course, we have many kind of protein and peptides that we are using in our day-to-day life, and there are many kind of autoimmune disorders and all these things in which we are taking these peptides and proteins, and many works are also done on this one. So like for this example, if I will take like improving the physicochemical stability, that is desmopressin and analog for vasopressin with the non-natural amino acid substitution for improved half-life and stability. So you can see that once uh, they are substituting it, and they are improving the half-life, and they are improving the stability and so on. Okay, and then if you take this one, the example that is non-invasive administration, that is a insulin human inhaler powder, that is Afrezza, and inhaled insulin powder consisting of microparticles formulated with formaldehyde, uh, diketopiperazine to achieve suitable characteristic for inhalable delivery. So here these days we are also working on insulin powders and all these things. So you can see that when we are trying to administer it with the microparticle, the formulated with fumarate D-piperazine, keto D-piperazine, that it is suitable for delivery of inhalable insulin. So this is how the degradation of the insulin can be stopped with this thing. So it is one of the way we can deliver the insulin non-invasively. That is because, of course, these days the insulin is directly delivered through the injection, and of course, many people are paranoid because of the injection and so on. So that's why this insulin can be delivered through the inhalable using a microparticle, and microparticle can be formulated with many other vehicles also, not only formaldehyde, diketopiperazine, but many other things also are used, like a suitable polymer and so on. Okay. Then we have antibodies. That is, antibodies are predominant class of therapeutic, mounting to more than 500 ongoing clinical trials and more than 70 clinical trials approved in United States. So you can see that how antibodies are being aggressively studied, and how many clinical trials are approved already. So the structure of antibodies, which differ substantially from that of other class of biologics, allow for a specific interaction between therapeutic targets and the immune system. Like antibodies provide signal to immune system by binding to the cellular targets. By binding to the target antigen, antibodies can neutralize it, preventing signaling molecules from binding to it and initiating the cell process. Okay. So here we have some examples and what are the challenges that it is being faced. It is facing, that is non-invasive administration. If you see for the antibodies, that is trastuzumab and hyaluronidase, that is a subcutaneous depot with the hyaluronidase for the sustained release of herceptin. Okay, so these are some of the examples for this one, that how the antibodies and what are the challenges of antibodies and how they have modified with the example. Like then nucleic acid, although the protein and peptide therapeutics have greatly expanded the number of druggable targets, nucleic acids enable the precise control of gene expression and hence be used to silence and repair aberrant gene and to drive expression of therapeutically active genes. So specific binding enables by their nucleotide sequence, nucleic acids, and more recent gene editing, such tools as CRISPR. I think CRISPR is one of the very known technique that we used in this time. And I think many researchers from University of Malaya also are studying on CRISPR technology as well. So can be rationally designed to therapeutically manipulate the human genome. Okay. So these are some of the examples for this one, that how the nucleic acid is being used, and like assessing the acetol and nucleus for following uptake, that is particeran, that is ionizable cationic lipid mediate endosomal escape following the endocytosis. Then we have the preventing of target gene editing, that is CRISPR CCR5 modified CD34 cell for HIV. That is the CD34 cell, the are edited by ex vivo, and whole genome sequence performed after editing and after engrafting defect of target effects. Then we have live cell therapies. That in live cell therapies are the newest generation of therapeutics. Live cell therapies takes advantage of natural therapeutics function of some cell type to regulate or enable key biological processes. Okay, and then in this one, this is for if you see the example, like what are the challenges? The challenges for controlling unpredictable PK parameter, that is preclinical alginate-based biopolymer implant for the controlled release of CAR-T cells directly to the site of action. So here what they are doing is the live cell therapy is that they will prepare a scaffold, okay? They will prepare a scaffold, and on the scaffold, they will put the live cell, and that live cell will migrate to the body and it will do their function. So these are some of the things that being used as a live cell therapy and so on. And now we'll cover up this three paradigm of drug delivery. That is the three core paradigm of drug delivery that span all class of therapeutics. So in this one, all class of therapeutics comes in, like drug modification, microenvironmental modification, and drug delivery system. Whatever you talk about in the delivery system, they will match to the one of the three paradigm. Okay, so we will cover up them one by one. That is the first is the drug modification. That is the modification of the drug encompass chemical alteration to its structure. Okay, such as the functional, that is the functional group, that is amino acids, nucleic acid backbone, or its conjugation to known moiety or targeting ligand. The purpose is to modulate interaction between the drug and molecules, cells and tissues in the body, as well as between the drug and its target sites, allowing for some control over how the drug navigates and proceeds within the body for initial administration down to its intended function. Okay, so I will give you some examples, or we can cover up some examples of drug modification. So here the category of the class that category or the class of therapeutics we have, that is the small molecules. So like the first modification done to achieve our target or to overcome the difficulties. Okay, so what are the modification? That is the modification of the functional group. So I think this is one of the very basic modification that is done on the small molecules to make it more soluble, to make it more, less effect of the pH and so on. So this is a modification of the functional group, that is the ritonavir. That is, it is a, this is example is just the analog of another drug. Okay, so it is being termed as a ritonavir after changing the functional group. Then masking undesirable chemical group, that is benzazipril. So all these examples are there. Then another example is the pegylation. What is pegylation? If you see in this one, the small molecules, the last one, pegylation, that is nalvoxigol. That is, so pegylation is that it is attaching of PEG. What is PEG? PEG is a solvent, or PEG is a polymer that is have very high property, and it can give stealth effect to the molecules and so on. So this pegylation can increase the blood circulation, and it can increase the blood circulation of the it can increase the circulation, blood circulation of that particular drug. Okay, hereby increasing the solubility of the molecules. Okay, then we have peptides and proteins. So there are also alteration of amino acid sequence, then alteration of intramolecular bonding, addition of non-natural amino acid, that is desmopressin. Then we have chemical backbone and modification, that is like cyclosporine A. Then we have pegylation, pegadimus bovine, and all this thing. Then we have antibodies. So what type of modifications are done on antibodies? That is affinity, maturation, and variable region engineering. Then antibody humanization, modification of FC binding to a neonatal FC receptor, and so on. Then we have nucleic acids and live cell therapy. So here also we have some kind of modification extending from genetic engineering to surface modifications and chemical backbone modifications. Then we have micro environmental modification. Then micro modification to the environment encompass a spectrum of approach. approaches from highly targeted changes at the site of action to the systemically administered adjuvant that alters the human environment. Then we have environmental modification represents a broad drug delivery strategies that can aid in navigation of biological barriers. For example, small molecules, excipients were developed to enhance drug solubility in physicochemical fluid by altering the local pH. Of course, there are many kind of small molecules which have difficulties in being released to the pH 1.2 because of course when you take a medicine the first pH that you face is 1.2 and that pH is very hard, so majority of the drug can degrade in that drug like pH 1.2. So that's why we prepare a tablet or we prepare a specific tablet which can go and release at pH 6.8, that is our lower pH. Okay, so these are some of the modifications that are done and it comes under the micro environmental modifications. So here are the examples of the micro environmental modifications and what are the modifications that are
I'm
done.
sorry, Dr. Syed, we are
Yeah.
almost short of time. So is it possible that we could speed up
Yeah,
the
yeah,
process?
yeah. Maybe two more slides. I will just try to finish it. Yeah.
All right, thank you.
So we have a class of therapeutics that is small molecules again. So this class of therapeutics are common in all, and so we are here different of examples. So I want you to focus on the modification. That what are the modifications that are done? Okay. So in the small molecule, the addition of solubilizing excipients, inhibition of clearance pathway. For peptides, we use of protease inhibitor and use of immunomodulator and so on. Then same goes for the antibodies, that is use of enzyme inhibitors, use of permeation enhancer, use of immunomodulator, so on. Okay. So these are the modifications that are done to achieve our target. Okay. Same goes for the live cell therapies also. In live cell therapy, use of signaling molecules to support the in vivo cellular action. So the last one is the drug delivery systems. Of course, these drug delivery systems are very vast and there are so many class of drug delivery system, that is modification to the drug to its microenvironment can adjust and optimize the activity of the drug. Drug delivery system can combine these two strategies by building an interference between the drug and its micro environment. So, okay, what are the drug delivery systems that we have here? Okay, I think I will use this one. That is what are the class of therapeutics first, and then the controlled release capsules. So these are the systems. Okay, here we are classifying the drug delivery system and the modifications or the shift that have been done on them. Okay, like controlled release capsules, controlled release implants, and all these things. So, and then inhalable devices, transdermal patches, microparticles, nanoparticles, targeted drug deliveries, and so on. Then we have peptides and protein. That is, again, it goes for the controlled release of microparticles and depots, targeted drug delivery, and in situ forming polymer matrix, implants, and so on. Then we have antibodies. Uh, and then we have nucleic acids and live cell therapy. So all these things are being done. So for nucleic acid, you can see that recently we have this in a vaccine that is lipid-based nanoparticle. Actually, what is mRNA COVID-19 vaccine is just a lipid-based nanoparticle. Okay, it's been made by a lipid structure, and the RNA is being incorporated inside it with other excipients for its stability and modification. Okay. So these are some of the things that being used in the modification or the in the drug delivery system. So these are like polymer conjugates, targeted ligands, and so on. So challenges and conclusion analysis of the existing landscape for therapeutics and delivery approach, drug modification and environmental modification suggests that there are three general outstanding challenges, that is the targeted delivery of the single cell or cell compartment resolution, overcoming biological barriers that limits the delivery of complex therapeutic molecules. Then we have the development of drug delivery system that rapidly secretes biomolecules in specific tissues at specific time and concentration in response to the environment cues. So these are the references. So the main reference is the evolution of the commercial drug delivery system. If you want to read this one, you can go there. And then just all about our faculty. And that's all. Thank you, everyone.
Thank you, Dr. Syed, for a very good presentation. So we're happy to take a question from the floor. So currently we have one question from Prof. Yu Quen Chan. He asked about what parameters are used to have slow release of drugs that just need once a month injection. Do they give consistent levels daily over one period?
Yeah, wait, stop sharing. Okay. Yeah, Fauzi, if you don't mind, can you repeat the question again?
So the question is, what parameters are used to have
Yeah.
slow release of drugs that just need once a month injection? Do they give consistent level daily over 1 month period?
Hmm. 1 month.
Yeah.
Oh, I think for 1 month, I think it's
For
not.
example, like
I
insulin,
know.
so they kind of
Yeah.
like give you 1 month kind of period, or like kind
Mm-hmm.
of like
I think for the yeah the thing is that for the injection deliveries and for the very direct systemic delivery actions and all this thing, the research is still ongoing, and maybe you can say that still we don't have these kind of therapies in which we can deliver an injection that can be used for 1 month, I think. 1 month, right? The question
yeah.
was 1 month. Yeah, I think it's 1 month is not possible. Maybe one day is possible, or maybe one dose is possible. Like, there are something called lag dose or overdose or something. So I think it's not possible for 1 month. I have not read such paper that say that the injection can be used for 1 month, I think. So basically what we are doing, even for the injections and all this thing, we are using like conductive polymers and targeted polymers, or the polymers which can react specifically to the tumors, or which can specifically to a target site according to the charge and all this thing. We are using those type of polymers in injections as well, or we are using some kind of especially designed polymers for this thing. But I don't think so that there is something which can be used for 1 month and so on.
Prof. Yu Quen Chan, he mentioned here in clinical practice, drugs needing only 1 monthly injection are already available.
Okay.
So perhaps,
1 month. Okay, okay.
yeah. So it's like,
That's
for example, like Pasurta. So there's a 1 month injection for the subcutaneous study. So perhaps you can elaborate more on this kind of drugs type
Mhm.
of delivery.
Uh, where's the question? Question is there. What parameter we
In
use
the
to
chat
have a,
box, please.
huh?
In the chat box, Q&A.
Yeah, yeah.
So you go there, there's a one question from, two question from Prof. Yu Quen Chan.
Uh-huh. What parameters are used to have a slow release of drug? Just need 1 month. Do you give any consistent? Uh, what I think then we only need to modify the polymers, or we need to see that what type of disease are there. Then only we can follow such regimes, I think. Yeah.
Because he already mentioned here that in clinical practice,
Mm-hmm.
drugs needing only 1 month injections are already available. So
Okay.
lead to the previous question, he wants to know more about what are the parameters for slow release of drugs that can be used in the drug delivery kind of productions.
So what type of parameter? I think we need to see the physicochemical properties of the drug, totally the chemistry, like if the drug is suitable for in vivo, is suitable for direct release, or if we need to see the bioavailability of the drug, like if the bioavailability is less than 20% and all these things, then only usually we are going for the direct injection drugs. Otherwise, if the drug is already 50% and more, I think we can go for the tablet and all this thing. But I think if the bioavailability is less and the solubility is less, then only we are going for this one.
He asking more about, he more interested whether there is consistent release.
For the consistent release, I think we have to go for the formulation modification rather than we have to choose the drug parameter, because the drug is fixed already. So I think we need to go for the drug loading, how much, what type of vehicle we are using. We are using which kind of vehicle, like we are using microparticle, we are using nanoparticle, we are using a transdermal patch, or we are using, I mean, like we are using what type of delivery system. So according to that, we can load how much drug, and then only we can decide that how much drug will be released over the time. So I think this is related to the drug modifications and so on. The drug delivery system modification as well. So according to this, we have to choose the polymers, and according to that, we have to design the formulation well. So there are very extended release patches, and there are extended release medications are there which are being used for three or four days. So why not, if there is something present for 1 month, or there is something present for 6 months also? I mean, it's an achievement. Yeah.
All right, I think that's all the time that we have now. So if Prof. Yukan can have more questions, we can directly contact to Dr. Syed for more
Yeah,
elaboration or
sure.
collaboration, perhaps so. So for the time now, so we are we're at the time limit. So thank you everyone for joining us for today, and then thank you for all the participants for giving us the pay attention to the Dr. Syed Mahmood's delivery today. Thank you very much.
Thank you, Fauzi. Thank you, everyone.